So today a few bits and snippets on just the genomics and what you can do with it and so on. So this is a whole list, but you know, it will be not as bad as it seems to look here. A couple of things you already know. Okay, let's start with a slight introduction. So ever since mankind was there and developed a conscience, they felt that mankind is a very special species. You know, we're all aware that we're so very different from other animals. We're smarter, we're more scientific. And the more you study, the more you find that at least higher apes do pretty much what we can do in all respects. But anyway, so when genomics was there, we could really start counting all the genomic stuff, the number of genes or the size of DNA. People were thinking, we will be so special. And none of this is really true. We are mediocre when it comes to the length of our genome. You know, water lily and salamander are 30 times bigger. And then, you know, people thought, you know, we will be saved in terms of the number of genes. So that must be, of course, fast. But then again, just mediocre. And so here you see also something interesting here on this slide, which is a bit older. The number of genes is 23,500 roughly. And now we are more on 19,500. So that means something, right? So we thought, so how much do we really know about genes? Well, you just, you know, refine the number by roughly 3,000 genes that happen not to be there. So still, understanding all genes and how the mechanism really works will need some considerable further studies. Okay, so let's move on. Oh, and here is that number, you know, to show you that we're mediocre. I think it's slide you might have seen before. You know, really halfway with this. Yes, larger in genomic size than bacteria, certainly the viruses, which typically, of course, are the smallest in the number of creatures. Well, let's call them creatures anyway. And as you can see, in a mammal, for example, laughs at us. We are ridiculously small, just an animal. So who is special here? Okay, I've alluded to you already about 16S RNA as a good marker to do genomics, to find out about where organisms are placed along the phylogenetic tree, and how to build these you've learned in the last couple of lectures. So it's Carl Lewis, again a bit of this. And yeah, so you know the nice thing about this molecule is that it has constant regions that make it reliable all these sequences and also variable regions. So that's very nice. And it's in all bacteria. And you know, if you go find a mitochondria and eukaryotic cells, you can also find 16S RNA genes there that you can use to compare with, for example, bacterial 16S RNA sequences. You've seen, right? And if you look at this tree, you see these two major events, bacterial origin, a green and a blue arrow. I think I'll show that a little bit. Mitochondria, you know, out of bacterial origin, alpha proteolytic bacteria that were once went into a predecessor of the eukaryotic cell, and that was a symbiotic relationship. The bacteria felt nicely protected by the whole environment, and all of the things that were excreted by that bacteria were nice for the host cell, so that remained in place. Even till today, we still have mitochondria as the main energy providing entity for each of the cells. Okay, I think also quick revision on horizontal gene transfer. You know, so it's acquiring material, that's the elevator story. Already. So here you will find 3 biological ways in which this can be done by bacteria. So they have you know, really elaborate systems built to transmit genomic material. And the main form of DNA in bacterial species, by the way, is a circular form called plasmids. Yeah. In our case, if we get back to this lecture, we have linear DNA. These guys do circular DNA. And maybe there's an advantage having circular DNA, but that back to that. Okay, so what's important for a bioinformatician if you have these things exchanged? It of course depends very much from which species to what other species it is transmitted. So as a result you can get compositional biases of very strange species, or you're doing an analysis and you find, oh, this whale is very similar to something else, to bacteria. If you're looking at the transmitted piece, they call it opposition So you have to be careful with analysis because basically genomicists and geneticists hate two things. Horizontal transfer, which screws up analysis and is difficult, and the other thing is sex they hate, but I'll get back to that. Okay. Yeah, all of this you can read it, because it is still real. Okay, let's do something else. Why do geneticists hate sex in their studies? That is, one of the applications that people would like to have, and it's becoming ever more popular, and most probably, as a Christmas present, you could consult 23andMe as a company and get a Christmas reduction for a genomics kit to trace your ancestry and so on, and really find out where you're from and so on, how much Neanderthal DNA you have on board. Numbers can go up, by the way, was 17%, but don't be shocked. Recent studies indicate that Neanderthal was a very intelligent species, and probably a lot more intelligent than the humanoids in those days, so nothing to be ashamed of. The percentage is a bit higher, but okay. So this is about, so what is the beauty about mitochondrial DNA? One of which is originally the origin, but what else? Who knows something? From whom do you receive, why is that?
Because you don't get the mitochondria from the sperm cell.
So man do not provide a lot to the world, right? With an egg cell, with energy, everything on that. Sorry, again, some people think it should be fake news. Anyway, so there's the cell, the egg cell, where everything is on board, and we just the sperm cell inserts just some DNA. Very little other entities. So most of it is just of maternal origin, and among this is the basic, one of the basic organelles in the cell, the mitochondria. And because it has DNA, that's very nice, because, I told you why it didn't hate sex. The nice thing about through the lineages, you just need to take care of the variability of this DNA, how much does this mutate, and the nice thing is that mitochondrial DNA mutates faster than our nuclear DNA, normal DNA if you like, and that means, you go through the lineages, you see differences coming up faster, but you don't get these mixtures that makes it very difficult to recognize what is time and what is, through the side nodes. And that is, of course, what you get with recombination and all effects that you get as a result of sexual transmission. So that is that. So, you can go through maternal DNA and, there are all kinds of kits around. If you want to try another one, you can do another thing. This is maybe a little bit of a sad story in a sense, because this is the one with Y chromosome. By now I hope that number 23 of the chromosome pairs is X and Y is the sex chromosome. So only one of the pairs out of 23 will determine the sex of a species. And but if you look at the karyogram, I've done the lecture on what was it? Genomic lecture, and the CNVs and so on, have karyogram for showing pictures by light microscopy of all of these chromosomes, and they receive the X chromosome, this, the Y chromosome, it's very small, very crumbled, and it's crumbling on. There are now even people who think that in 2 centuries, you won't see that anymore, but then the Y chromosome might be crumbled so much that it will not exist anymore. So I would say, put a few on this DNA somewhere, and you have just one male, where it will be a great society. Think about it. Maybe that's all for you to decide. I do all this stuff. Okay, so anyway, the Y chromosome is also nice in the sense, does it mutate so much? Not as much as mitochondrial DNA, but at least you know where it comes from males. Now, if you're a female, you might say, now we're in trouble because we don't have that chromosome, but then you can ask a brother or your father or whatever, or even a faraway cousin might do, because these Y chromosomes are really pretty stable. So you can just pretend that it's your own. So that's also what you can do. And normally these testing, and also what is happening, by the way, in court, as genetic or genomic evidence for somebody, a culprit who might have committed a murder, it's not still based on whole genome sequencing, none of them, just a few plants, and it's mostly repetitious regions, and also the Y chromosome. It's a repetitious tandem repeat, that gives even a line sort of color, and then the measure in a cheap way and it's related to all the size. So maybe that can be done better. So far, the test, but then they tell you that you're 70% Neanderthal. How do you find out that it's actually quite wrong? It's a way that the test is available. So, you know, and also thinking about this. So, my sister did a test, presuming that, you know, I'll be close to her. So, I just thought, you know, I'm just Frisian origin, you know, having A names, Dutch names ending in an A are normally from the north, right? To the effect that it was sort of funny when I started working a while ago at EMBL, 30 years ago, I think. EMBL is the European Molecular Laboratory. I was a great place. But then there was a big molecular biology lab in Groningen, which is kind of famous, a lot of people. So normally there was, you know, people from Groningen that would go to Apple. And all these names, for some reason, had ended in an A. And I came, I was not from Groningen at all, but my name ended in an A. So they were absolutely sure that Dutch names always ended in an A. That's not totally true. And somebody came, and I will tell you that story. They changed the whole setup of that ID in many respects, as I tell you. But okay. So this is about the which you can do. So I'm sure you can get this for cheap, 70 euros, 50 maybe by now. You can get a kit. Big swipe. Somebody smart. See where you get. Okay. Then, you know, just a thing that's of course intuitively clear in a sense, you know, how precise will you measure, and then they say if you have an 18 marker chromosome test and this is compared, then you can guess in between 1 and 27 generations, it shows here. And if you use a bit more markers, 46 is the example here, then you can, you know, trace back the origin into 1 to 10 generations. Still the grandson you have to include or something. But anyway, on the evolutionary map, what is 10 generations? 2 centuries, shall we say? Roughly. Right? Okay, a little story to hammer in evolution, I think. So we'll do this along a story from the Second World War, from the UK. We will talk about, you know, mostly the transcription affected binding regions and so on. So little motifs in DNA that have an influence on the genomic processes and regulation especially. So, little story then from the Second World War. England was quite active in the Second World War, fighting the Germans in those days. So they had a big air force. And, but so some of those planes would go over Germany, Bonn, some would be shot down, others came back, and they said, you know, the question is, how can we reinforce these planes as little as possible, because we can't be too heavy. But, you know, we get fewer casualties. So Abraham Wolf was a mathematician, and he was asked to analyze the patterns of bullet holes. And you know, first they thought what to do, and they done tests, and it hadn't helped. So here's the story: they got planes back. You know, some planes wouldn't come back, but some planes would, and some planes would be considerably damaged when they came back. So this is one example. You see where the bullet holes are indicated. And they said, oh, well, you know, that's not good. You know what? We'll reinforce these regions where the bullet holes have been. So they, you know, would reinforce the wings and the back. So the wings were important. And then, you know, the number of planes coming back didn't increase at all. Something went wrong. So then they started asking Abraham, well, can you have a look? I always told you, even if you don't know anymore, ask a mathematician. Even that was already known in England in those days. It doesn't work, ask a mathematician. So Abraham Wolf got back here. And he started looking, and he started reading a bit on Darwin and evolution and changing and what is important is conserved and so on. You know. So what's wrong here? What's wrong with the logic? It's totally wrong, I can tell you. It's absurd. Which planes don't get back? What do you need to protect? The planes that do come back, or the planes that wouldn't come back?
The ones that don't come back.
Yeah, and so what can you then say? It's like playing chess. Is this the biggest problem? The planes came back. So why do you think that the places you would not want to be hit are? Well, where is that? Where is the pilot? That's a good point. Where is the pilot? It's not with he or she. He's in the cockpit, he or she. Let's say he, to say the world. Okay. Where else? You know, there was something, a guy where the guy with the machine gun would sit. You know, don't shoot that guy, maybe. So, what about the engine of the plane? that is it. That's maybe not a good idea. Where would that be? Well, my guess is, seeing this picture, roughly there, in the middle. So what is it? So that's what. So we have this genomic ID here. So the plane is DNA right now, and mutations are the bullet holes, the bullets, and then there's functionality. So what do you see? That the real functional regions should not be hit by evolution. They shouldn't do that. You see, also here, Darwin is always right, even according to this mathematician. So, Everbold suggested that, you know what, I suggest you reinforce that middle area and something under the cockpit. Seen from the data side, right? We have many of these planes, we have those pictures, so it's a nice little student internship, you can always say, right? But you know, luckily we don't need to work on planes. We have enough DNA to do this if you want to do something, okay? So that's the main story. What is important is to know what is important, you should not touch and change. Yeah? Okay. So we're going to talk a little bit, and I've shown you, I think, a few species. Just the example paper will be a study that was done in the 2001 or something. Will be here somewhere, I guess. So a while ago, anyway, on 4 genome species where they really could find out a lot that was new at the time. Just looking with comparative genomics techniques at 4 different species sequences, DNA sequences of yeast. So here are the 4. The most well-known one is the top one, right, Baker's yeast, which is used in beers and so on. So, a very schematic representation of those 4 genomes is here, where you already see in red that a lot of things are really well preserved over these 4 genomes, but you see in blue a few regions, where you see that in those regions, which are larger regions, right, it's a whole genome, basically, that some considerable changes still can And so here you get also an inkling of another thing that's called synteny, where you could conserve a gene, but also what sometimes happens in a region where there are multiple genes with a certain order. If you preserve the order of the gene, so conserve the order if you like, it's called synteny. Yeah? So and if you see in related species, synteny is an important thing for which you can draw many lessons. So if the order of genes change, you know that some major rearrangements are taking place. Now, let's look. So they compared, and then they said, okay, we look at Saccharomyces cerevisiae, the Baker's yeast one, and we compare each with the 3 others. And we just count how many, in those alignments, how many nucleotides remained identical and how many were changed. Can you understand that this was the observation? In the coding sequences, comparing Baker's yeast with the 3 others, you see 90, 85, and 80% identity. What do you see typically? It's twice as low in a sense, or the difference is twice as high. 10% in paradoxus goes up to 20% different. 50 goes up to 30% different. So differences grow with a factor of 2. Is that clear in your mind why that is? This is the plane, right? The genes are important regions basically. So typically you find that there is a lot of observation. And the intergenic regions are more amenable to change. It doesn't matter. So you see, you can just count, you see it coming up in genomes if you compare. Yeah? Lesson 1. So, second observation. If you compare conservation over complete positions, over multiple alignments of the 4 species, then you see this happening. And again, and now you see also that there are 3 ways of counting mutations, and here is a gap mutation and a frame shift mutation. What's a frame shift mutation? That is just that you see in multiple alignment, gap, region, 1 there, or 1 there, 2 there by the way, that are of a length of unequal 3. Yeah? Because then, of course, you have 3 in those codon length, so you have gap 3, that might be the insertion of deletion, but the frame will change. If you have a 2 or 4 or 6 or so on, then you get a frame shift. And you see frame shifts will change the protein product dramatically, you know, and you see that in intergenic regions, the number of frame shifts is 75 times higher. So you see considerable selection against the occurrence of frame shift mutations in common regions. Yeah? And you see that the normal identity is a normal mutation, and then a mutation leading to a gap, so an insertion or deletion, is also doing more, you might say, because also there you see that it's more selected against 10 compared to 2 and point mutations. Okay. So then it did something else. It started looking, okay, so most methods until then were not really including comparative measures and of course mostly conservation. So they made a few sort of, they call dummy methods to just include thinking about conservation and including that in the methods. And one of the things was gene identification. Now, what is that? So for example, we all know how a gene starts with a start codon, and how a gene stops with a stop codon, and how would you think, and then they started thinking, and I think I've alluded that to you. There are, for example, when you think about stop codons, there are 3 stop codons in the codon table, 54 possibilities. 3 are stop codons. That means 1 in 20, roughly, randomly would occur if you have a random sequence, and that means most genes are much longer than 20 positions. So that means there's considerable selection against the occurrence of stop codons. Right? And so it's an easy mark. So, to cut the long story short, here you have something for this. This was known as a gene because it had ATG as a start codon. So this was taken as a gene earlier on, and they looked at this from this perspective of 4 yeast species with a line out here. And green, for example, is conserved region, so you might say certainly towards the end, it's nice conservation, and that's why they thought before this was a gene. But then if you look at the red is bad business, and also we can find out that even the stop codon, the last position there, is not totally conserved over 4 species, and that is a strong indication that this will be the gene. Right? So this was written off, not good. So it's a simple test, just like that. So you can see if you really include Darwinian thinking, get much further to better definitions of biological function. So, based on this relatively simple piece of research, they really revisited the gene catalog because a lot was known about linkage in those days, and they rejected a number of genes, found new ones, and changed the location of genes to start stop coding for 5% of the genes, so 1 in 20, but 6,000 genes is just not 20, it's just to find a number. And so also what they did is they confirmed in their new studies things that were known, and where is that? The red bit is what they did say. So what was known was 50, so they confirmed 30. They did not confirm 20. It's the study. And then they roughly in the percentages, they discovered 40 more new genes, and they never know what to do. What does that mean? That is clear. Some people would like to hope, certainly the method makers are for it, of course, it's in the back, it's biology. No, mostly it's not. So you really need to confirm and to validate those predictions. So checking the validity with really test results is really good. Okay. So they did something else, and that was about an investigation of the GAL4. That's a transcription factor binding site. We can show it in a moment. So it's a short, I've shown you that alignment, but it doesn't matter. So there's a regulatory motif. It's 6 to 15 base pairs in length, and it's pretty difficult to see in genomes. So they took this one as an example and thought, how can we prove that what is a real transcription factor binding site and what is just a fortuitous one? Because if you have such short motifs, 6 to 15 over the length of 2 billion letters, that human DNA is, and yeast is a bit short, not so much, then there is of course a lot of chance, high probability that you get these motifs just not having a biological function. They're somewhere in DNA that they can't be used. So how can you discern the one from the other? There again, of course, conservation comes to the rescue. So they tested that, and what they did is they took GAL4 motifs. So they said, okay, we know it's not totally true, but then there was more to believe. Transcription factor binding sites happen outside genes. So mostly preceding the gene, but sometimes following the gene. Preceding to following the gene. And so that's what they started comparing. So they looked at the GAL4 motif. How many of those land within a gene region? So that would be fortuitous one, when it lands just before or after, that might be a real one. And then to compare, I told you always need a null model, so they took they randomized the Del4 motif, and they started looking at those, and then they said, how many of the real ones do we find in gene regions as opposed to non-real gene regions, and how many of these random ones? And what happened, what they hope happened, so yes, you see that the random motif happens fewer times observed in the gene region and more often in the gene region, right, in the coding region here. So there was a difference of 1/2 or 8 factor 8. So you see that if you take conservation, that's a good way of rescuing, if you like, motifs where you would normally not know are they really functional or not. That wasn't good. Okay, so that was nice. That was all in this one paper. Made into Nature, by the way. So this is from our own studies where we made a model on the bottom there. And you know that there is a difference in bacterial genomes. You normally have one transcription factor, very few per gene. In the eukaryotic species, normally, typically, more transcription factors are involved, where some are binding to the DNA motif, but there are also proteins that are involved in these processes and do not bind themselves. The tiny stuff there. So the constituents of that multi-protein that together constitutes the switching box regulatory unit for a given gene. We start modeling them. That should be another story to know about. Okay, here you see from the study the 4 genomes and the GAL4 motifs they found, and you can see that there are a number of places where they're there. So that's also nice that you get also redundancy at this level, right? If that is important, you would have one motif that says some protein would bind at that moment to DNA, and we know they do. When proteins do bind to DNA, then it might be covered up, can be used. So typically you see multiple locations for important transcription factor binding sites to make sure that there's a high probability that they can be effective, it can be used. You see the Tata box, that's the 2 directions. GAL1 is a gene in the 1 direction, in the 4 direction, GAL10 is a gene in the reverse direction. And you see all kinds of frame shift mutations and so on. Right? There's one thing that came immediately out of the study. They had established that the GAL4 motif was CGG and then CCG, with some stuff in between, and that notion was already violated in this paper immediately. We see one of the longer GAL4 motifs in the 4th block from the top. You see that there is not quite what it would be. So that consensus 2 times 3 positions in one. And it's still a proven GAL4 motif. Anyway, biology is always comes. Okay, but at least from this study, they indeed found a lot of new elements using these conservation and finding some likely regions, some of which in the meantime, or most of which in the meantime have been performed experimentally. Okay, so that was an easy sort of study to do, just check for the first time. That's why you get an important landmark paper. So they found new motifs and a lot of knowledge was added just using the notion of comparison. Okay. And that's just let's talk about something else. And I already mentioned to you that bacterial DNA is mostly circular and that we have linear stretch DNA. And what is the problem? Maybe I told this to you already at an earlier occasion, but I'm not sure. Think about shoelaces. What is the annoying thing with some shoelaces? If you're in a hurry in the morning, you need to run out of your lace. It's not in your shoe. You have to put it back. Which are the annoying laces where the plastic cap is not there anymore, so you have to do this. It doesn't work, but it takes time. That's also. And that's also very important in DNA, I'd say, because these ends should be preserved. If they start crumbling, then it means DNA is crumbling towards the middle, and if there are all kinds of important genes that will be affected, that's not a good thing. So, like laces, what the DNA needs to cap. But then, when they made it, evolution started becoming smart, and they start maybe thinking, and that was evolution will think. Well, if you look back, it is as if it's been thinking. But whereas what was developed was a time clocking system, counting system to know how many divisions a cell has undergone since that DNA has started being there for the first time. And that's an elaborate system where we learn a lot. And typically I can tell you that cells in our body will die after 40 to 50 divisions. Then the cell itself will decide, I'm not good anymore, because there's always a chance, of course, build-up of error. It's all the time DNA division at each cell division, so this goes pretty well, very high fidelity, but still errors come in, so they will be added, multiplied, and so on. So, therefore, in evolution, typically cells do not get much older than 40, 45 cell divisions. So you need a counting system. How can a cell count? How often it has divided? That is done by means of these telomeres. So here, what do you see? This is a picture from this telomere paper, where you see that about mutations in DNA. So you see these colors. You know, don't look in the detail, but I hope you agree with me that at least at the ends and the starts of this DNA, you see a lot of colors, also sometimes in the middle. So that means here and here a lot of stuff happens. So it's really an area where evolution is active. What can happen? Let's just look at slides. So telomeres are basically our genomic is a DNA region. And typically what you find DNA when it stops like this. So this is DNA, the double one, right? And typically what you have is this. So telomeric region might start here. So normally of course DNA is double-stranded, but then the end, you know, one sticks out a bit more than the other. And the last couple of years, this region here has become important. Also, it is better against cancer. So what is happening? So this is what telomeres are. You have, so most of this is there's a lot of proteins playing a role binding in these regions. There are also specialized proteins that bind the single-stranded part, the very end part of DNA. And these proteins need to protect because normally what happens, DNA really starts crumbling fast if you don't take any protective measures. Okay. So here you have the story about how genomes became linear, starting from circular DNA. So here telomeres are complicated machines, told you that already. And here is the important thing. They regulate the lifespan of a cell, because how do they do this? At each cell division, a number of bases are chopped off the telomere tree. The telomere is short. And the cell has a measuring system to measure that short. And after 40 to 50 divisions, apparently in most cells, the shortening of the DNA from the telomeric end is so much, so pronounced, that then the decision is taken to that the cell should die. Right? And this is programmed cell. Apoptosis. That's called programmed cell. So the cell dies. Now, how do these telomeric regions look like? They have very many short repetitious elements. Here you see a couple in the second bullet. You see those in different organisms, vertebrates, it's TTA, GGG normally, and in bacteria it's very often TTT, GG, 4Gs, stress something. And so, these repetitions are numerous. Human telomere, you can have easily 15 and even 20,000, people told me, of these bits of sequence repeated. But at each division, and that's interesting, at least 50 to 500 of these repeats are taken off, are lost. And if there's enough time, if you multiply that by 40, then the cell will decide, okay, I'll go into apoptosis. Then, there's a nasty disease called cancer. And so this has to do with aging or anti-aging, right? Cells, so it utilize the system in place. But what is cancer doing? And that's one of the big problems with cancer. There is a special enzyme, Telomeres, that is able to elongate the telomeric region. So on one end of the cell expects it to get short, now it's too short, but in the cancerous cells, these bits are again elongated. So the decision being taken is too short, we die, is not happening. So you get these old cells that live on, and will make all kinds of effects. Of course, at 85, you get trouble in the end, because you didn't equal for nothing. So that's a big problem. So a lot of the battling against cancer, learning about the cellular networks, what goes wrong, is also just zooming in into the telomeric region. That is interesting because something happened there, and that has to do with, they found it from bacteria, many of these things sticking out, really, the single-stranded bit, very often have 4 Gs in a row. And then something interesting can happen, and that is seen here schematically in this picture. There are proteins that bind to it, and then what you can have is that at the very end, these telomeres, so do it like this. Here's one, and here's another molecule of DNA. Yeah, like that. Okay? So, now you have the protein complex. I think this is even a 4. This can go up to 4 copies of DNA that can be taped together. So you have got a protein complex. So the requirement is you need 4 Gs in a row in each of these DNAs. That is a signal for these proteins to bind. And then you might say they can take 2 strands of DNA together, or even 4. But what is the side effect when that happens? That the telomerase, that the elongate, the cancerous effect, cannot take place anymore. So if this happens, then the telomerase, producing cancer if you like, cannot bind anymore. So this makes, this is one of the maybe possible solutions, or at least strong things. So now a lot of pharma, a lot of focus is placed on this. So, you know, although it's mostly for bacterial origin, and develop proteins, although the cell has this effect, you need to find out how bad it is if this happens anyway, of course. Is this, you know, without all these flapping bits, are these DNA still functional? You hang off this. So there's a lot you need to research, but it looks up. So there are some good results. So let's hope that is something. Okay? Now, a few more general things. This is about the telomerase, just, you know, and also here, of course, comparative genomics is very important because it can be variable. So you really need to find out how things are concerned. And that was the subject of the study that I showed you a few slides about. So, phylogenetic footprinting. Just a very general technique. The catchphrase here is what is important is conserved. So how does it work? Here's a picture. There will be more than 1,000 words. So you see those in green. These say these transcription factor binding regions. And the top box, and then in red the genes. And you see that sometimes, you know, like in the yeast species reported, but should be really conserved across the 4 species. Sometimes you have that you get a variant, TFBS prime, TFBS2. Sometimes it's not totally conserved, and so on. So you get differences, and this, of course, you know, gives some understanding about the importance of each of those binding domains, binding regions, binding motifs. So basically, there are all kinds of techniques to do that. So basically, you could do it online with alignment or alignment free. We had a lecture on motif searching, so you could run mean, find some motifs, unaligned sequences. Normally that is used, these techniques are used to trace transcription factor binding sites. And here you see an example of technology where, you know, you see alignment would be a difficult thing to find those. Also because, you know, the black lines in the alignment is very dissimilar bits of sequences, so alignment is hard, and the motifs are normally very short, even if they are totally conserved, very hard to align. So techniques that do not need alignments come in handy here, and here you see an example of some of the regions. In yellow, the color coding in the sequences is the same as in the picture at the top. And Mathieu Blanchet has been one of the champions in phylogenetic footprinting, so this way to find him. Now, we do the reverse side of the coin. What is important is conserved. Another technology, in addition to phylogenetic footprinting, is called phylogenetic shadowing. That is about resuming not on the things that remain the same, but on the things that become different. So here you see an example. The top sequence is the human sequence, and we're interested. What can be changed in the human sequence if we compare those sequences with those sequences you see under the human sequence, so mostly chimps here, spider monkey, so these are all mice. So basically it is as if you, okay, there is a long strip of light on the human sequence, okay? A mutation is a bit of a wall, you see that here. The red is a bit of a wall. So the light tries to shine through, but it's, of course it's stopped at each bit of wall, and you capture in the end, you have a screen, and you capture what light will touch the screen. So you see there that most regions are stopped, they don't emit any light anymore, but the screen, you know, where you still see light is these 3 regions where an arrow points to, and what regions are those? These are the regions that did not accumulate, that did not show any difference in any of the other species. So what are the important regions? The white ones, but in the shadowing. So, this technique zooms in on what can be different. So what is not important can be different with the catch tracing. And the red bits here in the picture. Okay? So this technology was used to find out about related species. These were apes, all of them. And they could with this technique they can pretty well time the moment where something splits. So this tree is pretty reliable in a number of years. So then you see what we are, what is our nearest species, the chimp, right now. And you see these other ones. So we're also pretty close to a gibbon. So I don't know if you ever watched the gibbon. Anyway. Okay, so here you see that. So the idea is that these techniques can only be used for closer sets of sequences, right? And if you use it for material and theories, then everything will always have different inquiries and translations. Yeah? So I've talked about the binding sites, the transcription vector binding sites extensively now. And here is a database, it's called TransFac. In fact, you should take the slide off because what has happened, normally you often find that databases get funded if you make a new database. So you go to your national funder and you say, we have this fantastic database, we would like to make a database of super factors. Because they're really important for a lot of people. And then the committee support it, so they have to be dedicated. Yes, funding. So after 4 years you get back and say, it's a great success. A million people use our database. We have to maintain it. Can you give us money? Oh no, this is no money. We have no money. So that means the lifetime of the database is always hard. And then what do? You play this and you get no after 4, 8 years, whatever. No other money anymore. You go private. You close the gate. People have to start paying. The transfer is mainly gone now. And then you take a risk. It will be important that people still use you. That's very often not the case anymore. Because scientists are still beasts. And there's no money to pay for this, they do. So anyway, then people look again at some free possibilities and so on. So that's a bit of a problem. So this hit also TransFac. There are many databases, even Swissquote. Who has not heard of Swissquote in this class? You should not dare to stick with the free. Okay, when was that? 2000? No, 1996. Swiss product, the Swiss government would fund it, and then Europe would fund it, and then as well they had something very nice. The Swiss said, you know what? We impose a new rule. Anything that's funded by Europe doesn't need funding from us. And then all the Europeans did the same. What is funded by Swiss doesn't need to be funded. That's called deadlocking the computer website. And they were waiting for a while. There was no funding. Who came to the rescue the next day to tell you? US, can you imagine? Well, very good, they did it. And that is a database that if it's not up for a week, a lot of research is going to collapse. Really. It's one of the mistakes of the database. And even there it happens. So it's hard, databases. No issue. Okay, so this is the database, and what you see is just very simple. You might say this is a profile with just the numbers of nucleotides you find in positions. So the length of the motif is there, this one is 14 long, and just the count of the nucleotides that occur at each of those positions. Okay. SNPs, single nucleotide polymorphism. You know what that is? And it feels like a mutation, right? It is a mutation that happens enough to be counted. So if you have 10,000 sequences and there's only one letter that is different, you would probably not call that a SNP. It's too risky, probably a sequencing error or something. So SNPs should happen enough. So they should happen not frequently, but frequently enough that that will count. Right? So that's basically all of these points. Look at that. And SNPs are very interesting because for cancer research, genetic diseases, of course, we would like to know what diseases are affected by mutation, if there be any relationship with the disease. And that's what people are doing. Great numbers in the databases, in hospitals and in companies. So SNPs are, of course, a way to compare sequences. Well, you get SNPs by sequences. Okay, so this is an older slide. So how many SNPs do we have? I see different people sitting here. You know how enormously similar you are at the genomic level, right? You know how many in how many letters is different between you? Here is too high. It says here 1 in 1430. It's 1 in 1,000. It's a bit more. But still, it's a line, right? We people aligning DNA is not in most regions. Now, based on all these SNPs, they were looking, and if it's a cancer, for example, then it might be interesting that you get it through your maternal gene or through your gene from your father. So there is, in most of this research, these things come in place, and what people always hope to get in terms of data is triplet data, the DNA of the patient, plus the DNA of the SNP of the father and the mother. If you have those triplets, you can really start researching and work more effectively. And that is, and that is about the phasing of SNPs, of what kind of chromosome, and why is that? If you see what happened, what's the difference between possibility 1 and possibility 2? What should have happened before? So the top figure is GG from the father, let's say, and TA is the genome of the mother. You see the difference, what must have happened? What happened in the region 1, possibility 1? There was no crossing over because both SNPs from the father are in the child and from the mother. That is the child, right? So here you get possibility 2 parents would be like that, then you must have crossover in the child. That is interesting because crossing over is something that geneticists, classical geneticists use to find out how correlated some genes are. How often is there crossover in between? And that is how they start doing it. So there's a whole theory about it. This is sort of classical genetics. If you start thinking about it, if you have a gene A and a gene B, a region, and they come with 2 alleles, 2 forms, 2 alternatives that are a big difference. So big A and small a for gene 1, and big B and small b for gene 2. So the outline kind of lines up like that on the genome. Now, suppose if they were totally independent, how would you think you would get the distribution of these alleles in children, in offspring, and they use those, the frequencies in which you find that as to indicate how correlated genes are. And if they will be totally independent, for example, A and B will be on a different chromosome, what kind of mixture would you then have in terms of, would you just get the random situation? Chance for the one, for big A, times the chance for big B. If they will be on one closed region, then if you have a big A, the chance is very high that you also get big B, because they're on the same strand. So if you do that, then this is the mathematical form you do. So your random motif, you have your random chances, if there would not be any connection, no correlation at all. So the chance for F big A, big B is just, it's all the product which you would randomly expect. But then, if they are occurring together on the strands of DNA, and the breaks are not so numerous, then you get something on letter D extra, as you can see, and that is normally mentioned called linkage disequilibrium. So that's the D comes from disequilibrium, and that means that it's not random, not there is a connection. So a frequency as a result might become higher or lower as a result. That is indicated by those D's in those formulas. And of course, to calculate, you see there that the real frequency of AB, you have to subtract the random frequency to get the linkage disequilibrium. So how can we use SNPs if you do it like that? So, the phase interference between the father and mother tells you where the SNPs are organized, where they are positioned in the genome, and then with this linkage equilibrium formula, you get a sense of the correlation between SNPs. And that is interesting if SNPs are correlated. And that is because of the next example, haplotypes. What are haplotypes? They are multiple SNPs. The question is that come in handy if you have multiple SNPs. So for example, you found out this one SNP is really important for the disease. But the problem is you can't measure it. It's close to the central ring of this motor sequence. You know it's there, you know it's important, but you can't get your hand on it. But you know that there is another SNP that you can measure easily with a high correlation with this bad SNP. That's very nice if it correlates 80%. The other, and you measure that and do it in the little uncertainty, but 80% is better than nothing. You can start predicting which patients are in trouble, people are in trouble, or not. So that's the idea. So that's on this slide, a little example, where you have an haplotype. So this is just a lining up of SNPs. So this first sequence is just a sort of correlated SNPs. They could be over a wide stretch of DNA. So this GGA, there could be thousands of nucleotides in between. And so that this is the count you see, and there's a number of observations in this table. You see they give them names, of course, like A, happening in 83% of the cases. These letters line up, and that means if your first G is really correlated with the disease and the last C is measurable, you can use the C pretty well as an indicator, as a marker for that disease as well, because of the correlation. Right? So if the correlation gets lower, okay, stop it at what point you can. I must tell you in history, when this came out 10 years ago, haplotypes, everybody jumped on it because everybody was this is the solution to cancer. Now we see it. In the end, it has not brought this great relief that people thought it would occur. And but still, it's there. There's less a hype at the moment than it was probably back. But yeah, so haplotypes and SNPs, they're important cancer research. So by now, I think I've told you a few things that are related to the sense of comparing, right? Ever since Darwin, we know that comparing. As a biologist, you do a lot of reversals in here, right? You don't know things so much. The machine itself doesn't tell you. There's not in the cell something saying, I'm a liver cell. Say that. You need to look, find where it is, and look, and then there are different types of liver cells. And I stopped dividing because something happens. We need to reverse engineer all those things. And the simple story to indicate the importance of conservation is a Martian visiting this planet and looking at cars. We have various cars, right? We have trucks, we have racing cars, also cars. So the Martian looks at this and will say, circles under car important. We know that circles are also wheels. But why is that? Why could the Martian say that? Because there are virtually no cars without wheels. And normally the number of wheels is 4. In the great majority. So, and then, I don't know if that works so well these days, but a little protrusion out of the top of the car, antenna, is not so important. Why? Because not all cars have it. So, and those two remarks are absolutely right. Wheels are more essential to a car than an antenna. So why comparing you can find it? So that's the message, the main message that I'm going here. And all of these ways are, the things I showed you are ways to compare and to look at it, to extract meaning out of what you see, right? And it can be used almost everywhere. Also in planes during the Second World War, right? So evolution is a really important message. So by that, let's take a short break. Where are we time-wise? Okay, so we're half, so take 10 minutes and then do questions, okay? We have at least half an hour for questions. So during your coffee, start thinking about my class. This is your chance. Thank you. Say it again? No, well, if you like, it's fine too, but raising your hand and asking your question is good enough. And I'll repeat the question so everybody hears it. Yeah. Yeah? Why don't you state your name? That's nice for me.
Okay. I'm
Alice, okay?
With the multiple sequence alignment method, you make a multiple sequence alignment based on tree, which is supposed to be quick and dirty.
Yeah.
And then you use the lower bounding score.
That's you're talking about this method that's trying to to all possibilities. The MSA, that's the method.
Yeah.
That's what we're talking about. Yeah. So that is so that is not the most important protocol is progressive alignment, right? So this is not it. It's trying to do everything. So what they do is they do a quick and dirty alignment. So their idea is we want to not go to all cells in the search matrix. And in the programming, to check each and every one. If this is millions of cells, that takes too long. Write off as many cells as possible. So how would you do this? You say, okay, we use a standard alignment method that we would like to do better than that. So they come up with scores in the cells, right? The proper alignment scores like this, and they can just, so they do that. It's pairwise alignment, and then it might be that, you know, that maybe with a bit of luck, this is the region that the alignments that would fall in here have cells that score high enough according to some criteria. Yeah, so they say, okay, we do an alignment, and we only take those cells that score better than a given number. Given that bad alignment. You know, if it would be perfect alignment, then it would be different. What's your problem with that?
Well, they first make a multiple sequence alignment.
Yeah. No, they do all of this at the pairwise alignment level, at the beginning level, not during the multiple sequence alignment. Well, they could sometimes use a multiple sequence alignment, if that is there, but that's then a cheap one and a fast one to do it. Because this is extremely slow.
Okay, but then you would just make a fast one.
Yeah.
From the fast one determine we don't look at that.
Yeah, you learn from the fast one and then you do it slow in a better way as well.
Okay, and you don't have to understand how they determine which things they can do.
Well, no, it's very simple. It's, you know, so the quick and dirty alignment method gives scores, and you put a certain threshold, depends on what you learn there, you say, okay, we're higher than that, we keep you in the list. Yeah, and the rest you won't look at. So if that scores of these cells here, if all of this is too low, then you will not look there, so you can be a bit faster when you go through your alignment steps. Yeah. Don't worry, these methods are not used anyway. They're totally impractical. Yeah? Okay. Another question. Yeah?
Yeah. At some point you mentioned that there are two...
State your name.
Okay, then yes.
Yeah.
You mentioned that there are two sums of their scores.
Yeah.
And they are different dimensions. But how
Yeah.
do we know which one you're talking about and what is the actual difference?
Well, this is your alignment score, you normally calculate in your pairwise alignment thing, but your main score is a pair score. And that is a score based on an alignment. So it's one alignment. Now the other symbol pair score is when we're comparing our query alignment, the alignment we made with the program, to some reference, and then, you know, stating how good you are, evaluating how good you are. So that is another, it's just the same thing, that's annoying. But, you know, then you talk about the sum of pair scores resulting from a query alignment, your alignment, so to speak, your method alignment, and some reference alignment, or two alignments you want to compare in that sense. And you could also use sum of pair scoring to compare two methods, to see how the alignments look like one. That's the difference. So from the context, you can easily see what it is. But still it's another. And by the way, in bioinformatics you find this all the time, right? The same title for different things, different words for the same thing. And if you're in the database industry, you have to sort of this ambiguity. That's called in the Wikipedia as well, right? Finding out what is what. Okay. Another question? Not really? One more? Yes?
So in neighbor joining there was the example, and then we had A, B, C, D. So first we took A, B as closest ones, and then while continuing, there's like a new calculation, a new matrix.
Yeah, you do these conversion steps. So don't worry if in detail you don't get it.
Okay.
Think about how it works if you compare it to UPGMA clustering. So it chooses what to add to the tree in the end, right? This neighbor joining thing. So overall, so you start, you have all single sequences at that moment. They make this fake tree, so you have a fake point in the middle. Right here. Sequences, the distances, you take the average point and then you know your distances from the sequences. And now you start checking all possible pairs. And you check if I do this, if I could take these two together like this. Right? And this is the distance I'm talking about. And now, so that's then a new situation. So overall, pairs, you look, and why would you choose this one over merging these two pairs, or this one and that one, for example? That is the total tree length is little more than this. As you can see. So that's why you need all the distances. That's why you need that fake tree in the beginning. And then these conversion steps are done. And then you have your new branch length. And then it's just doing that again. So with that, what happens is, if this has happened here, it will be 30 anyway. So this is a new situation. What it does, it says, okay, I take this point now, which you can calculate, and this, I don't look at, that's on the sideline, of course I have it. But now the next situation is the tree now has one entity less, right? And then you do the same thing. So it could be you merge this one with that, or that one first. So that's the picture that's on the slide where this happens. So in the end you have an unrooted tree. So, but I'm not going to bother you with really the formula and how to make it goes. And if I would be so criminal to do this, I will give you all knowledge you need. So I'm not going, you don't need to remember formula. I will give it to you. You only, in fact, need to really memorize the things I told you before. You have to remember that, of course, first, and then you remember what it was that you need to remember. Remember that, they are okay. So I'm not going to tell you, as the 4 steps of multiple sequence alignments progressively are this, because I told you you should know that. And the ingredients, the 4 things you need for evolution, if you don't know that, should be a tiny bit ashamed, so go and learn. I won't give that to you, you should know it. Yeah. Okay. Another question, yeah, that's fine.
So in the meme method, we have the hidden state problem. Hidden state problem. So we are doing the expectation step and maximization step. But I'm not sure if we can start without probability matrix for the expectation. So we need a random or like prior knowledge based one.
Yeah.
Okay. And then from that it will send.
Yeah, exactly the same as what you do in your hidden model. You need a prior. Where you get it from, good luck. If it's a smart prior, you would get to, you would converge to good results sooner, and all the time you need to test all the possibilities to be sure. You walk over the landscape, you try to get to the highest or the deepest value. That's always the truth. There are other methods, by the way. They're called simulated annealing. That happens in as just, so you know that is like you could think about that as if you raise the temperature, so you have the landscape, and you would like to find the deepest value. So you can do the cumbersome thing of yourself walking down. But now you raise the temperature, that is as if you now take a helicopter. You fly over. And then, you go over and you take bigger steps in this direction, and then you go down. So then you have a higher chance to land in deep value. So there are all kinds of physically inspired methods to try and optimize the search. We put it in the market, that would be a bit too much, because this is already showing all the issues, and that's okay. Yeah. But yes, that's so you need it. Yeah. Yeah.
In the test exam, you asked for the complexity of.
Yeah.
I was wondering.
Which one was that? The multiple alignment complexity? The 2 steps?
Yeah, multiple sequence. Yeah. And first of all, I was wondering if it's an all against all, you compare, for example, sequence 1 to sequence 2, and then also 2 to 1, or is it only 1 to 2? That's okay.
And one in alignment. You program it. Would it make a difference if you have sequence 1 and 2 on top? No. So you need to do one of those. And how many comparisons are there? That's a formula you might know off the top of your head.
I thought.
If it's half a matrix minus the main diagonal, how many possibilities are there? N times n minus 1 divided by 2. That's the number. So that's the number of alignments you do. Pairwise. Yeah. It's like football. Ajax, you know, in football you have the out game, the home game, the out game, right? So there's double. But here,
Isn't it like the same as in faculteit?
no, far more, far more. Faculty is exponential. No. This is. You know, this is the number of sequences all against all, right? Sequences here, sequences there. So it can never be more than n squared. So it's less than that. How much less? It's still a square relationship, but it's less because you know what you said, A to B is same as B to A. That means if you have the number here, you don't need to bother. So half this matrix is out. And now you tell me, what about the main diagonal? Do you align sequence A to A? Is that interesting? No. Okay, so we don't need that. So how many cells are left? The under triangle minus the main diagonal. And you think a little bit, and then you go to n times n is the whole matrix, minus n for the main diagonal, and then the left half, and that evaluates if you do that to n times n minus 1. Yeah, so that's the formula.
Oh yeah.
And if you work it out, do you know what you get? N squared minus n, right? Divided by 2. So what's the bad news in terms of scalability? It's still square scalability, whatever you do. Eh? It's a bit less than just n squared, but it's still a quadratic relationship. Yeah? And that is so the question there is, you know, why do we want that? Why do we do all against all in progressive alignment? Let's talk about that. Why is that? What do we need? Why was that?
You need a distance.
Yeah, but why do we need the distance? What do we do in the end with it? After step 3, how do you use step 1? Remember, all against all. Step 2, similarity matrix. Similarity matrix is step G3. Then step 4 is the alignment, so that's the important step, you might say. What do you use? Step 3, what is then the tree? And what is the tree? It's an order. You use it as an order. You don't even use these branches like everything to calculate. You say, okay, these 2 guys first, and then that. You follow the tree. So you need a tree to follow. Order how to do this. How much do we put in to calculate that? Well, all against all alignments, you know, clustering program, a lot of work. So how many steps do we have? Well, these number of computation steps when you have n sequences, at least. And then when we have the order, the last step, step 4, that was the question in fact. How many computation steps do you have there? Is that quadratic? The real alignment step, step 4, using the tree to make the alignment, is n minus 1 computational steps. Look at the tree, here you have a tree. There's another one like this, so how many objects? 5. How many splits do you have? You see the relationship? It's all n minus 1. So it means linear in the number of sequences when you do the real alignment, and quadratic in the number of steps when you do the preparatory phase of getting the order. So anything, so a lot of people reason like, oh, is the ordering so important? No, not really. So let's do something cheap. So I could ask you, as an advanced question, give me something fast that is cheap and also gives you order. What is the cumbersome thing you're doing when you're comparing all the sequences? Use bloody sequence alignments, for example. All this matrix and calculating and worrying about scores. So can we do this quicker and dirtier? Don't worry. What can we do? Let's give you something. What is a protein sequence? It's a collection of amino acids, right? How many different are there? 20 times. So, if you now would just forget about the order of the sequence, how many are linked, how many are in there? You do this for 2 sequences. Calculate the distance. What do you need to do? Compare 20 numbers. That's quick. So quickly to read through the sequence, count, put the factor, and then go around as to do that. Multiple is a method from the 80s. My old boss in England later. So he said, okay, you know, I just assume that the order is not so important. So then you can do something quicker with it. In the Pauline method, I started making this double. No, I think that's terribly important. So I zoom in on this and make it even slower. And hope to get to squeeze something out. So that's the fun about making methods. All kinds of people do different things. It's all about cracking good alignments and making them reliable. But what is important? To try things. The messages that are somewhere contained in evolution, you should respect and follow. But then, you know, evolution in one corner of life might be, although it's all Darwinian perhaps, different from others. Hey, you know, these families that are widely divergent and all the families are, you know, very, very similar. So the rules are very different how you play, right? Size of the ball, football, if you have kick or not, and, you have offside, and all these things will change. But it's all still football. Right? Okay. Yeah. No, please.
Continuation on this question.
Yeah.
You would also have to use the length sequence, which you can
Yeah. That will be in. So tell me.
L square.
Yeah.
That's it.
So give me the complexity of the one now. Now you're aware. What's the complexity? Of the first three steps, so the three generations. What complexity is that? Yeah, this is not good enough. This is too precise. It's of the order of, right? So what would your complexity be? All sequences have length L. Order of, what's this? N squared, thank you very much, times? Yeah, why not? There you go. And the order of the little step, so to speak, the alignment step is? Yeah. That's good. And here, of course, you know, it's mostly profile, and of course, comparing two profiles is a bit slower than, you know, in practice, this is of the order of the number of steps. Yeah. So that would be the answer. But you see that the preparatory phase, so to speak, in the progressive strategy is more expensive than squared than the thing we're all on about. Okay? That's why I said, what about the method that you take? Because the sequence is in order. Don't worry, order is not important. So I do the first and the second, and then I align to the third, and then I align to the fourth, you know. Quick. I will do that. Okay? Anything else? Now, if you don't ask questions to me, I will ask questions to you. So, quick, let's go. Quick question. Yeah.
I was wondering about the programming that you use in defining that penalties.
Yeah.
There's like,
In fact, the fine penalties are the normal death penalties.
Yeah.
In the sense that they're used by yeah, but you mean linear, right? That's normal.
Yeah.
Yeah.
Because you have this line where you have like the word maps.
Yeah. The GoPro algorithm. Yeah.
I don't understand those words like for the algorithm because in the normal and programming you do that three cells.
Okay. In the normal algorithm, you what do you do? You're in a cell, you can go from three ways, as you know, right? From the top, from there. Okay, and that means if you would follow like this, that means a gap, right? If you are here and you come from the top or the left, do you know how many gaps you've had before? You don't. And we had this trick, like if you want to go to a file, in fact you only need to know whether the gap is opening or in the extension phase. And that meant you look at that cell back and then you know. It's a bit of doing things and then you can rescue that one. But if you want a general gap penalty, let's say we make a scheme of gap penalties here, I make one. You can say it's biologically maybe nonsensical. Here we go. You can go on like this, so the gap of length 1, 2, 3, 4, 5, and 6. And this one I make 10. And you know what, this one I make 11. Makes sense, right? But now, this is a very strange biological case. I got an 8 here. I don't know why. And this is 15. And this is 100. Strange function, right? Very wobbly. You know, normally we have the linear is like this, right? And the affine goes like this. And then I told you about this concave one, that is something like that, but we didn't touch that yet. Now, suppose you would like to have that one. In your standard error, you can't do it. Because you would say, if you have a general gap penalty scheme, any crazy gap penalty score, given the length of your gap, if you would like to include that, then in fact you need an algorithm where at this moment you know where you come from and you know the number of gaps. So that's this GOTO ID. So the phasing is different. Here you can be on the way in a gap. Coming from here there, you can be busy doing a gap, right? In the GOTO matrix that doesn't happen. Any cell is always a match-mismatch. But then if you have the funny horse drift like that, and you would come from here, so something like that, then you know, okay, here's where I come from, I jump to here, so I have 1, 2, 3, 4, 5, so the length is 5, right?
I don't understand how the why are you gaps checked like above the cell? Because that comes with that
So this is how you evaluate the GOTO algorithm, the preceding column leading up to the cell diagonally, and this. That is precisely for that reason. Now you can only come into this cell from these, so not directly to the left anymore or directly from the top, because that is this possibility in the algorithm that you're used to, where you're during, so the phasing is different. This is after you're done with the gap. Any value in the cell is after you're done. Otherwise you wouldn't be able, if you're during the gap, you can't tell how long it is, how long we go on. The algorithm can only say things in the past. So that's why in the cell the definition of GOTO is after. And that means then you need to do it like that. Because now you know exactly, if you come from here, you know how your jump was. So in the old algorithm as you're used to, you know you walk like that and then this, and the gap is that long. And then you can say, okay, what was the score here? At that moment, and now I put in the score and I put the gap penalty for the gap of 5. Maybe not, and then you know that. So you know exactly. So you can now use crazy gap penalties like that. And not in the normal algorithm. How long you've been here, a bit of hanky-panky. You can rescue the what you yeah. Does that answer your question? Are you not talking? Yes.
I don't know what it is.
All right. Get back to what you have. Yeah. Anything else? Anything else for the weekend? No. That's a general question. Very good.
Can you repeat again how the first step of class works? Because
First step of class. Okay.
we have like streamers,
Yep.
then
Yep.
we take similar books to that, and
Yep.
it gets.
Let's try. So we have three sequences. Whatever, right? First step is take the 3 words, right? 3 words. So each of those 3 words, let's say this one, we look in the 8,000 possibilities. So this is what the 3 word GHI say. We make a list of things that are close enough, all the 3 words that are considered close enough. We will use a threshold value for that. And what do we do? All the 8,000 possible words, 20 to the power 3, so 7999 then in this case, because we've got this one. We check them all, and you know, the first word will be, well, AAA. So if you would enumerate them, they go like this, right? So all possible 8,000 would be D, and so on, right? So you just check them all. And what do you use to compare? You don't do realignment, you just look at this, it's a gap plus alignment of 3. So these two, you are testing those. You take the blossoms values. So the GA value, the HA value, the IA value, you add those 3 up, and that gives you a number for this thing. Right? You do this for all, and then you take a threshold. You should score over something. So that is the list. So for each of the 3 words, you will build a list that has a subset of the 8,000 possible words, depending on the threshold. Yeah? And basically what you now do, how do you use that? You have your matrix. You know, the BLAST is a sequence searching method. So you have a query sequence, let's do it in the BLAST way. It's here, and your database sequence is, sorry, the query sequence of course is here, this is mathematical now, and this is the database sequence. So you're all about using this to find the best sort of these diagonals, right? Where are local areas of similarity? The FASTA method that I also explained really looks at exactly the same letters in the one sequence and the other. BLAST uses more virology, more divergence if you like, can capture that by this trick fast. So this basically will allow that some of the letters GG, A might qualify as a word that is close enough. So when it does that, it looks at the 3 words, and then, okay, now for these 3 words in the query sequence, yeah, that was in the list, so it does the thing. This is for all the words, and here might be another one in the database sequence that is doing that. So perfect. And then, elongates when the next word would also have something. This is, so the end you have all these sort of small diagonals, and then you get the to-hit method, and all this to work it out further, and then the full alignment. This is still the quick step, right? And and then MAST has this automata to make this really fast, so that you immediately know it's indexing in the end, and you know immediately if you go through the database sequence, you know immediately where these hits are. So you see that, okay, check the sequence. So that means for each sequence the treatment is linear to the length of the database sequence. And normally, of course, if you would just do alignment, then it would be quadratic again. Right? So you work back. So that makes it faster because of the automata they use. And preparing all of this, massaging the query sequence, doing all of this might take time. But that's only once. Okay, you can do all the searches over all of the databases. I think this index rate. The same happens for BWA, in another way, that's the secret. So they build the big indexing tree and all of that only once, can be quite expensive, but then you do all your alignments. Okay. Anything else? Burning questions? Okay, let's go through. What did we do with this? So I gave you a bit of biology, not too much I hope. Here's the people who come without biology can still keep up. I hope it worked for those who didn't know anything when they came to the course. I think we started with alignment. Local, semi-global, global, in the clear. You know where the cell is to be found in the global, you programmed it up, so I hope you remember that. Yeah? How it works, philosophy. Okay, this order, you go to that is one thing, don't worry too much about it. I don't think I'll ask you questions on that. Think about something that is more general, I might ask, I'll give you the stuff that you need to answer that. So, that was alignment, we did blah, we did multiple alignment. The main method, of course, we have questions. The progressive alignment strategy, and this exhaustive method that was asked about. These are not used in practice, too slow. We did blast, of course, we have questions on that. We had one question on the motifs. We have the EM algorithm. What did we do? Last time, any questions? Anything else? That was it, right? Oh, and of course the trees. Did you ask about the trees? The first tree. Anything there? You asked them, right? Well. Yeah, this is true, but yeah, no, you're right. Of course, we did a question. Very good. So we have questions about all subjects. That's good. Anything else? So if you have a burning one and you really feel uncertain, you can always make a new one. Don't do it on the night. I'll be out. I promise, but sometimes I don't feel it. So give me a bit of it. All right. Now, I enjoyed doing scores with you all. I hope you enjoyed it also. The practicals were nice enough that you felt it helped. I think the students in the sense I see how they were learning about doing all of the things, sending announcements. So I hope that you felt that too. If not, tell us. You'll get your questionnaire, right? Be honest with you. Don't be negative, don't be positive, be honest. Okay. Thanks.